Good evening, everyone, and welcome to the American Society for Microbiology Microbes After Hours program. We're thrilled to see you all here tonight. I'm also excited to welcome our online audience from Microbe World, our ASM branches and student chapters, and our ASM educators who are watching online. My name is Shannon Green, and I'm the American Academy of Microbiology Colloquium Fellow. The Academy is the part of ASM that produces FAQ reports on hot topics in microbiology, such as influenza, the human microbiome, the microbiology of brewing beer, and will soon include a report on this very topic, the microbiology of cheese. ASM is a society that's over 100 years old and has almost 40,000 members, but those numbers are absolutely nothing when compared to the 4 billion years in which millions upon millions of microbes have dominated life on Earth. Microbes are everywhere, microbes are essential, and microbes are extraordinary. And if you were able to attend our last Microbes After Hours in October or watch the video online, you already know that microbes are essential to the production of one of our favorite beverages, beer. And here tonight, we have two fantastic speakers who are going to take us on a journey through the lifespan of another favorite microbially produced food product, which is cheese. So I'm delighted to introduce Matteo Keeler. Matteo started Jasper Hill Farm with his brother Andy in 2003. They produce a wide range of cheeses from the milk of their herd of 45 Ayrshire cows. In 2008, they started a new venture, the Cellars at Jasper Hill, a 22,000 square foot underground cheese ripening facility that lowers barriers for entry for new cheesemakers for maturing, marketing, and selling cheeses, managing logistics and administration, and providing technical support to local producers. The Cellars at Jasper Hill is committed to developing economic mechanisms to keep the working landscape in Vermont's Northeast Kingdom working, and delivering deliciousness is the core and principal component in this effort. Welcome, Matteo.
It's really a thrill to be here, and really I feel very privileged to be participating in the colloquium. So I just wanted to start by polling my audience here. I know all you folks out there in internet land won't be able to necessarily see this, but I want to be able to understand who I'm speaking to in this room. So if you're a microbiologist or involved in microbiology, will you just raise your hand? Oh my goodness. If you're involved in the food industry, will you raise your hand? And oh, look at that, huh? We got 3 lowly cheesemakers. So, are there any regulators in the room? I won't count you. Yeah. Great. Well, I'm going to try and give you all an overview of cheesemaking. Many of you probably know a lot more about cheesemaking than I do, but I'm going to talk a little bit about the fundamentals and the chemistry part. And Dr. Rachel Dutton is going to follow up with the microbiology. So from our perspective, as a farmstead cheesemaker, cheesemaking begins with grass. I think about what we do at Jasper Hill is sending cows out to harvest a patch of grass. Cow is a still, and we're basically sending cows out to harvest a patch of grass, bring it back to the barn, and we think of the cows as a still, right? So we're concentrating that grass, that sunshine, and preserving it. Historically, cheese has been a store of value. Microbes are fundamental to making milk. When a cow eats grass, when we feed a cow, whether it's hay or grain, we're not just feeding the cow, we're actually feeding microbes in the rumen. And cheesemaking is, a long chain of basically taking proteins apart, putting them back together, and then taking them back apart. So the rumen is filled with billions, hundreds of billions, trillions of microbes that are breaking down the fiber, the lignin, the protein, the sugars, and fermenting them in the rumen. And the bypass protein, the dead carcasses, the cells that pass out of the rumen, are actually what the cow digests, breaks down to produce milk in proteins in the udder. So microbes are a fundamental part of the cheese-making process right from the beginning. But what is milk made out of? Principally, we're looking at water. About 90% of milk is water, but there's also casein, which is possibly the most important component of milk, and that's represented by that couche ball there in the middle. And we'll talk a little bit more about protein in a minute. But that's a fundamental structure of cheese is protein. We also have fat, lactose, which is the sugar that is fermented by microbes to acidify milk. We have minerals, which play a very important role in texture. More minerals equals longer, more elasticity in a cheese. Less minerals equals shorter, a shorter texture. And so what we do through the cheese-making process and the fermentation of milk has a huge impact on the final product. One of the reasons I love cheese is that you start with the same 4 ingredients, but you can make 1,000 different products. It's kind of a miracle. Milk is home to a very wide variety of microbes, and these microbes come from a variety of places. In the barn, you have feed, manure, bedding, people, skin. 80% of the microbes in raw milk come from the outside of the udder outside of the teats of a cow. So there's a lot of work going into studying the microbial ecology of farms. Specifically in Europe, there's been quite a bit of work that's studying how farm practices create the microbial ecology on the teat end of a cow. And I know there's a few scientists in this room that have been working on some of this as well. And we've been really fortunate to have had the opportunity to have our farm and our cows sampled routinely over a while. And we're learning how the microecology of our own barn and cheese-making facility informs the microbiology of our finished cheeses. But it's a chain. Ultimately, the microbial life is collected through milking equipment. That's also a reservoir of bacteria, depending on how effective you are at cleaning your equipment. And it multiplies in a tank where the milk is collected. So if you're making a pasteurized cheese, that's your opportunity to basically sever the link between the microbiology that exists in your pasture, in your animals, the farmers themselves, and make cheese. If you're making a raw milk cheese, this is where that microbiota will multiply. So the first stage in cheese making is actually the addition, or if you have a healthy population of endemic lactic acid bacteria in your milk because of your practices, the addition of lactic acid bacteria, which metabolizes lactose, produces enzymes that help break down proteins, and there's a wide variety of lactic acid bacteria, and they are endemic in most areas. We've sequenced our milk and milk from all our producers and found indigenous strains of lactic acid bacteria in all our milk samples. But that acidification is fundamental to the production of cheese. So after you add your starter bacteria and those sugars are being fermented, the next stage is the addition of enzymes to coagulate milk. And basically, coagulation is the aggregation of casein micelles. Casein micelles are comprised of all the different types of protein that a cow or a lactating dairy animal produces. But the casein has kappa casein, which are the little curly like extremities there that are where the polarity. Let me back this up. The aggregation of casein micelles is accomplished through enzymes or acidification, right? And when you either have a lot of acid production or you introduce an enzyme like rennet, you're basically changing the polarity of these caseins. And instead of repelling, which is what they do when you have a cup of milk, they attract to each other and you end up with a gel. And that gel is what looks like that. So that's basically an image of a Bailey-Hazen blue set just after it's been cut. So it's a gel, and it's basically an aggregation of the proteins which locks in the fat, and it's a calcium phosphate, the minerals that actually form the bonds that hold these proteins together. So there's different types of rennet. There's animal rennet, which is extracted from the abomasum, the fourth stomach of an unweaned calf. And when I describe cheese making, basically what we're doing is we're replicating the way that a calf would digest milk. And a calf is, suckling on its mother. That milk bypasses the rumen when it suckles and goes straight to the abomasum, where it's coagulated. The solids are captured and digested, and the liquid is secreted. There are other types of rennet as well. There's cardoon thistles, which are traditional in some cheeses from Spain and Portugal, and there's also microbially produced rennet. And there's some controversy now about whether or not that is GMO or non-GMO, and I think that we'll be hearing a lot more about this moving forward as we start to sort out what a GMO actually is. The primary phase of coagulation is the clipping of proteins between the 105th and 106th amino acids. And then the secondary phase is the formation of these calcium phosphate bridges that actually form the gel. There are two different families of cheese, and they're distinguished by the type of coagulation of the milk. Lactic cheeses, which, like a lot of chèvres, for instance, are lactic coagulation, is basically lactic-dominated coagulation. Yogurt, when you add lactic acid, will reach an isoelectric point at about a pH of 4.7, and that's the stage at which you get that coagulation of proteins. Enzymatic coagulation is dominated by the addition of an enzyme like rennet. And those cheeses are going to be more like Emmentaler or cheddar, hard-cooked pressed cheeses tend to be enzymatic. After curd is coagulated, it's drained. This is an image of ladling of a lactic cheese, so a soft cheese. But if you're making Parmigiano Reggiano, you would basically be cutting your curd into rice-sized bits, heating, basically extracting the moisture and creating the conditions for ripening. So available water is a big part of the final cheese. The characteristics of a finished cheese are going to be determined by the moisture content, and a lot of that is determined by what you do at this stage, draining your curds. And then off to the cellars to be ripened. So just a couple of words about the cellars and what we do. The image to the left is the footprint of our building. We're about 30 feet underground. It's a 22,000 square foot underground complex of tunnels. We've got 5 different environments. We ripen natural rind cheeses that range from soft Bloomy rinded cheeses to Alpine hard cooked pressed cheeses to blue cheeses to tome cheeses. And our business model is really about purchasing cheese in from producers so that we can basically cash flow their businesses early. They don't have to carry the costs of inventory. And we take on a lot of the costs of ripening and marketing cheese for producers in our area. But what happens in the cellar? Then therethere's 2 approaches to cheese making, and one is bowling, where the trajectory of a cheese is set in the vat, and you throw a ball down the alley and stand back and see whether you got strikes or spares or you got a gutter ball, and you can get pretty good at throwing strikes and spares. But what we do is a little different than bowling. We're basically taking cheeses, using environments and selective pressure, different tools to select for different microbial communities on the rind, which affects the characteristics of the final cheese, right? So these are some of the tools that we use to actually ripen our cheese. You've got air speed and gas pressure. So how much oxygen do you have in your space? How much ammonia do you have in your space? What's the temperature? How long are you, we can move cheese from one space to another, but all these tools are basically intended to give us some control in selecting for different communities on the rinds of our cheeses. And I always say that you can't turn a donkey into a racehorse, right? If you've got a donkey coming in to the building, you're going to get a donkey coming out. Affinage is not magic. The trajectory of a cheese is established in the vat, and even before that by the quality of the milk going into that vat. But what we do is basically ensure that a cheese reaches its full potential by giving it the treatment and listening to what it has to tell us. And if you spend enough time with the cheese, it will speak to you. It'll be there saying, I really need to get turned over. Will you please turn me over or brush me or wash me? And in a way, what we're doing is cultivating these microbial communities the way that you would cultivate a garden, right? And that's really an exercise in consciousness. So I love my job and I love cheese, and it's amazing the variety and the mystery that cheese is. And I'll just leave you with one idea, and that is that cheese is a discovery, not an invention. So we're on a learning curve that's steep some days and long and subtle other days, but the opportunity to be involved in cheese at this point in time where we have technology that gives us the ability to see what the microbial communities that affect and comprise our products this is an incredible time to be in cheese. And it's my pleasure really to be here and to be involved with a lot of you in this room. 10 years ago, I described cheese as being like, being a cheesemaker as being like a god, except you're blind and dumb. because you're unleashing these universes of life and then like wiping them out and not understanding exactly what's going on. And now we have, we might still be dumb, but at least we have the ability to see what's going on. So it's a really exciting time to be in cheese.
Thanks, Matteo. So I know that everyone has lots of questions for him, but we're going to hold questions till after our second speaker, which is Rachel Dutton. So after receiving her PhD in microbiology from Harvard Medical School, Rachel was awarded a Bauer Fellowship at Harvard University to start an independent research group. She combined her passions of microbiology and food into a research program that uses cheese as a way to understand microbial ecosystems. The Dutton Lab studies the microbial diversity of cheeses from around the world and looks at how cheese microbes interact with each other to form communities. Rachel's been a speaker at events such as the World Science Festival and regularly gives classes to the general public on the science of cheese and other fermented foods. Research from her lab has been featured in Lucky Peach magazine, The Mind of a Chef TV series on PBS, EdibleBoston, the Boston Globe, NPR, and the New York Times. Please join me in welcoming Rachel.
So I first want to thank Shannon and everybody else here at the American Society for Microbiology and the American Academy for Microbiology for hosting and organizing this event. It's really a pleasure to get to be here with everybody, and I'm looking forward to the discussions tomorrow for the FAQ. So Matteo talked about this amazing diversity of cheeses, and in my mind, really what this represents is an amazing diversity of microbes, and in particular, communities of microbes. And as somebody who loves cheese, I see these cheeses and I think, you know, this is incredible, you know, world of flavor that we have accessible to us. But as a microbiologist, I look at these cheeses and think that this is an incredible universe of microbes. And every single one of these cheeses to me is like this wonderful universe that we are just starting to understand. So we're sort of out there looking at this universe and trying to make sense of how it all works. And I want to show you some images that we've taken in my lab from some of these communities of microbes, which I think will make sense why I think of this as a universe. So these are pictures of microbes that we have basically scraped from the surface of these cheeses. So we study the rind of the cheese, and this is what forms during the aging process. And we see this incredible collection of microbial species, and it's quite diverse. So we have examples of molds, so filamentous fungi, as well as many different types of bacteria, as well as yeasts, which are also fungi. So this is one particular cheese, which has an interesting mix of beautiful microbes. Here's another one that's particularly photogenic, different types of microbes growing together. Another gorgeous sort of starscape here of additional microbes. These are all different types of cheese. Another wonderful set of organisms. So this is just sort of a glimpse into our world as cheese microbiologists, so the sorts of things that we really love seeing and drive us to ask questions about how these communities form and what these microbes are doing. And besides cheese itself being incredibly fascinating and having a long and interesting history, one of the reasons that we're interested in studying cheese is that microbial communities in general are so important. So we're learning, you know, probably every single day in the news, another story about the human microbiome and microbes in other environments and why they're so important. And so we have examples from, for example, the soil, where there's billions of microbes in every gram of soil, and, for example, the human gut, which is just chock full of microbes. And so we're learning a lot about how ubiquitous these communities are, as well as how important they are for the health of humans, as well as the health for pretty much every ecosystem on the planet. Now, the challenge in understanding what is happening within these microbial communities. So we know they're there and we know they're important, but how does it all work? And part of why that's difficult to answer as a microbiologist is that these communities are really complex. So they have hundreds or thousands of species living together. Many of these species we don't actually know how to grow in the lab. We don't know how to exactly replicate the conditions in which to grow them. So that makes it really difficult to understand who's doing what, how they're interacting with each other within the context of these complex communities. So one of the goals of my lab is to try and find a simple system for understanding these communities. So something like a model system. So just as we use E. coli or Drosophila as model organisms, can we have a model community that might tell us the basic principles of how communities work? And so ideally, we would want something that is much simpler, something that's reproducible, that's made. These communities exist very frequently. They might be easy to manipulate to produce different types of communities and easy to get a hold of. So you might have guessed that I'm describing cheese. And so what we're doing in the lab is trying to use cheese as a way to really understand how microbial communities work. So we're taking cheeses that cheesemakers like Mateo at Jasper Hill makes, and we are completely tearing them apart in the lab and putting them back together and asking a number of different questions. So this is on the right here, you have a picture of 96 well in vitro cheeses. So we actually make cheese media in the lab, and then we can recreate a cheese rind on the surface of these different cheeses and manipulate them in different ways. So we're asking things like, how many species are there in these communities? How diverse are these communities from one cheese to another? And what determines what type of community you have? What are the manipulations that you need to do? How do these species interact with each other? Are the microbes communicating? Are they at war with each other? Are they at peace? Are they helping each other? How does it all work? How do these communities form? And I think by understanding these basic ideas, this might be able to tell us something about these more complex communities in other environments. So the part of cheese that we spend all of our time studying is the rind of the cheese. And I think this is a completely amazing example of a microbial community. So you have this community forming during the aging process of the cheese, so in the caves. And it's basically a surface biofilm on the top of the cheese that forms. And you can see this sort of thick layer, and it's this mixture of bacteria and fungi growing together. And the first time I saw this, it reminded me of biofilms that I had seen in an environmental microbiology class that I had taken, which are the microbial mats that you find in salt marshes. And so I show this just to make the point that the type of community that we see on the surface of cheese is not a unique example of microbial life. These microbial mats are the oldest fossil record that we have of life on the planet. So we have fossil records going back 3.2 billion years of microbial mats. So microbes have been living in these types of communities as biofilms for billions of years. So here's a up-close look. This is looking top-down at the surface of a cheese. You can see this sort of the top of the biofilm on the surface, and then conveniently a little chunk of this sample got teared apart during the sample processing, and you can start to see the microbial life that's within the rind here. And so now imagine that you shrink yourself down to the size of a microbe, and you're standing inside the rind of the cheese. And you look around, this is what you would see. So you see this incredible, diverse, and jam-packed community of microbes around you. So you have evidence here of different types of bacteria, as well as some evidence of fungi, and these organisms are all living together in this community. And it's very dense. So there's about 10 billion cells per gram of rind. So think about that next time you eat a piece of cheese. And these organisms are alive. So it's really interesting to think about what they're doing. And as they're growing on the surface of the cheese, they're having a huge impact on the flavor of the cheese. So for many cheeses, a significant portion of the flavor, the texture, the aroma that you get in the cheese is actually directly a product of these microbes that are growing on the surface. So these are just some fun examples. There are hundreds of different compounds that microbes growing on cheese make, and depending on which collection of species you have, you can imagine you have a diverse possibility of flavors and aromas. So these are some of my favorite examples. 2-methylpropanol smells like bananas. Methanethiol smells like rotting cabbage. Methional, like sort of roasting meats. Butyric acid smells like sweaty gym socks, which you may not think of as necessarily a desirable aroma, but some of these very funky cheeses, that's what you're smelling. Diacetyl is a sort of buttery flavor. Benzaldehyde is a sort of like bitter almond. So there is a huge diversity of the chemical diversity that is imparted by the microbial life on cheese. So I thought I'd go through just a few examples of kind of the superstars of the cheese microbial world. So a lot of the research that's been happening over the past 10, 15 years in cheese microbiology is showing that there's a lot of diversity within each of these species that I'll talk about, but also beyond these organisms that we commonly think of associated with cheese. So I'm not going to have time to really talk about all these other examples, but just tell you about some of the key players that we already know quite a bit about. The first one that I wanted to focus on is a fungus called Geotrichum candidum. And this is the fungus that you will see growing on the surface of cheeses like this. So this is something that is called a Bloomy rind cheese, and it has a sort of brainy or like ramen noodle kind of look to it. And this structure is actually the biofilm that's formed by Geotrichum when it grows on the surface of the cheese. In the lab, it grows as these pretty fluffy colonies. And I have a picture here of Sister Noella Marcelino, also known as the cheese nun. And she's actually here in the audience, and we'll be tasting some of her cheese. Some of you have tasted it already, that she makes at her abbey in Connecticut. And Sister Noella actually has done really the seminal work on Geotrichum in the field, did her PhD studying the diversity of Geotrichum in France. And so we owe a lot of our knowledge on the diversity within this particular organism and in terms of cheese microbiology in general to some of the work she did during her PhD. And if you haven't seen it, please watch The Cheese Nun, which is a PBS documentary about Sister Noella, which is fantastic. Especially if you're a cheese nerd. So Geotrichum is one of the more common fungi that you find on cheese. And when you go into a cheese shop and eat a cheese that has this organism growing on it, it's pretty distinctive the way it smells. And one of the things that Geotrichum is good at is secreting enzymes called lipases. So all of these organisms have many different things they can do. This is one of the things that Geotrichum is known for. So it can actually secrete lipases. So it's growing on the surface of the cheese, secreting these enzymes into the cheese that allows it to break down the nutrients that are in the cheese and use those as food. And in the process of utilizing those as food, it's producing metabolites, which are these compounds that can have very, very interesting aromas. So for Geotrichum, when it secretes lipases, it can break down some of the fat in cheese to free fatty acids, which have some interesting flavors. And then it can further metabolize these fatty acids to things like methyl ketones and thioesters. So thioesters can be fairly stinky, cabbagey sort of aromas are often associated with this particular fungus. So it's very interesting. I encourage you to try it next time you're at a cheese shop. In Bloomy rind cheeses, there's another very common fungus that's used, and that is Penicillium camemberti. So you can tell here that the microbiology came after many, many years of cheese making, since the microbe was actually named after the cheese that it was identified on, camembert. And Penicillium camemberti is a filamentous fungi, so it's the white fluffy mold that you see on the surface of camembert. We have a beautiful scanning electron micrograph of one of the canidiaphores, so the structures that these molds produce to make spores, which are sort of like the seeds of the fungal world. It makes these really pretty fluffy colonies in the lab. It's related, the Penicillium species are related to the organism that we get the antibiotic, which is called penicillin. In a room full of microbiologists, I think we should be able to get this. So it's a different species that makes this makes the antibiotic, but these are within that same genus. So Penicillium camemberti, again, has the ability to secrete enzymes into the cheese as it's growing. One of the things it's really good at is secreting proteases, which can break down the casein protein. And this does a couple of things. One is that it leads to the production of some of these very mushroomy flavors that you get in the cheese, but it also has a big impact on the texture of the cheese. So often with this type of cheese, you'll see just under the rind, it'll start to kind of liquefy. And that's because the organism is secreting enzymes, and so they're diffusing just a little bit in at a time, and you're getting breakdown of the cheese as this organism is growing. So the longer the cheese is aged, the more time the organism will have to secrete enzymes, the more broken down the cheese will be, the more flavorful, the more gooey, eventually until the cheese is basically running away from you on the counter. Okay, so another penicillium that's very, very important in cheese making is Penicillium roqueforti, again, named after Roquefort cheese made in the southwest of France. You can see from the microscopy that it has a very similar type of cell structure. It produces these blue-green pigments when it forms spores. And this particular organism is actually inoculated into the milk of a blue cheese. And so unlike Penicillium camemberti, which loves oxygen and requires oxygen for growth, Penicillium roqueforti is actually a microaerophile, which means that it only likes a little bit of oxygen to grow. So it's a little bit picky. So during the production of a blue cheese, once the cheese is formed into its shape, the cheesemaker will actually take metal spikes and poke holes into the cheese to allow just a little bit of oxygen, and then the blue mold will actually flourish under those conditions. And so it's a pretty labor-intensive process to do this for cheeses if you're doing it by hand. But as the blue mold grows, one of the things that it's really good at, again, is producing lipases. So in blue cheeses, you have some of these flavors that are very characteristic due to the production of enzymes by this mold. So Penicillium roqueforti breaks down fats to free fatty acids. One of the free fatty acids that's produced by this particular enzyme is the peppery, spicy flavor that you associate with blue cheese. So every time you eat blue cheese and has that sort of spiciness, it's because of the free fatty acids that are produced through the metabolism of this fungus. And then it can metabolize this further again, and this time it produces a compound which is called 2-heptanone, which is the key aroma that we associate with blue cheese. So every time you smell blue cheese, you know it's blue cheese. And part of that is because you're smelling this molecule that this fungus is producing. So I think that's completely fascinating, that you can link a specific molecule that we associate with something very familiar with a microbe. So another type of cheese is a washed rind cheese. And these cheeses, during the aging process, so Matteo talked about, the affineurs have these different tools that they can use to kind of usher the communities in different directions. One of these tools is washing. And so the cheesemaker will actually wash the surface of the cheese with a brine solution. It can be very frequently or sort of intermittently, depending on what type of cheese they're making. One of the things that happens is you create this moist, salty environment on the surface of the cheese, which favors the growth of certain types of bacteria. We're learning that there are many different types of bacteria that grow in these conditions. One of the ones that we typically associate with these cheeses is Brevibacterium linens. And you can see a picture of Brevibacterium growing in the lab. It actually has this orange color. And the cheese here is Oma, which we'll taste after the lecture. And Brevibacterium linens is interesting because it again secretes proteins, breaking down the casein to things like amino acids eventually, which can have very brothy, meaty flavors. So these cheeses often taste very meaty. The interesting and sort of different thing about what happens here is that you end up breaking down these amino acids even further to produce things like amines and aldehydes and sulfur compounds. So those are things that smell sort of fishy and garlicky and sweaty. So often when you smell one of these cheeses, it smells like the gym sock or the stinky feet. And it actually makes complete microbiological sense because Brevibacterium species are found on our skin and are actually thought to contribute to body odor. So they are not the same species, but related species growing on our skin as what are growing on cheese and giving you very similar types of smells. So hopefully that doesn't gross everybody out completely if you weren't already grossed out by everything I've told you. Okay, last one, which is one of my favorite ones to talk about, are the Alpine cheeses like Swiss cheeses. And so I think everybody always wonders where the holes come from in Swiss cheese. And unfortunately, it's not these adorable mice. They are adorable microbes, however, that make these holes. So Propionibacterium is a microbe that is really encouraged in this type of cheese making, and some of you tasted this cheese alpha tolman, which is this type of cheese that you have this sort of organism growing in. And what Propionibacterium can do, it's pretty cool. So after the lactic acid bacteria grow, they are fermenting the lactose and producing lactic acid. So there's lots of lactic acid still around in the cheese, inside the cheese. And there's not that much energy to be gained from making a living on lactic acid, but these bacteria can do it. They eat the lactic acid, they ferment it basically, and they produce a few products. One is propionic acid, which is one of the key sort of Swiss kind of cheese flavors that you get, kind of tangy, a little bit of acetic acid. And the key thing here is CO2. So what you can imagine is that you have the Propionibacteria inside the cheese. It's a strict anaerobe, so it only grows inside the cheese. And everywhere it grows, it's eating lactic acid, producing CO2, and it's sort of forming this bubble of CO2 around it. So every time you see one of these holes in a Swiss cheese, you can imagine that there was a Propionibacterium growing there. Not a mouse. So glad I cleared that up. Okay, so in summary, I think hopefully what we've been able to tell you today that cheese making is a complex and wonderful process which involves microbes at every single stage. And these microbes really completely transform the grass into milk, and then milk into this diverse and delicious assortment of cheeses. And I will end there, and thank you, and hopefully take questions.
So thank you so much, Rachel and Matteo. We can take questions now. So if you are in this room, I'm going to ask that you guys go up to the microphone, off to, I guess, my left, your right, so that way our online audience can also hear the questions. For people who are watching online, we are happy to take questions from you guys as well. We'd encourage you to tweet your questions to @microbeworld. And you can use the hashtag #AfterHours in order to do so, or you can also ask your questions of the speakers through the online chat room on microbeworld.org. And we have some microphones actually on the front table for our speakers. If they can come up, we can grab microphones for them. They can answer questions. Okay, so I have a first question for Matteo that kind of came to mind when you were showing the layout of your cellars. It sort of looked like architecturally they were set up sort of in different ways, and I was curious, are similar cheeses always aged near one another? So for example, if you set like a blue cheese to age near like a washed rind cheese, are the microbial communities potentially going to be able to intersperse between the two? Or do you always keep certain cheeses of certain types aging with their brethren?
Well, we generally try and keep different styles of cheese segregated to minimize cross-contamination, which saves us work, basically. But also because different environments really require, different cheeses really require different environments. For instance, if you're curing a washed rind cheese, you want a higher ammonia content. That ammonia raises the pH on the rind. Ammonia is alkaline. Ammonia is produced by proteins breaking down and off-gassing, right? And they build up in the environment. They can really be a problem for like bloomy rinded or mold ripened cheeses, and in which case, those environments we're going to want a much higher oxygen content, right? So the environment really impacts the development of the rind on the one hand, and the cheese itself impacts the environment as well. And so that's why we have the 7 tunnels. We really only have 5 environments, but that's actually sufficient to do just about everything we need to do.
Well, we're probably all cheese lovers, which is why we're here, so thank you very, very much. I guess my question would come from sort of an infectious disease standpoint, because probably like many of us here, we've always cut the rind off the cheese and discarded it. And yet we're all very interested now in how we can create a healthier, gastrointestinal tract with, microbes and all. So are you suggesting that perhaps the healthiest part of the cheese should be the rind, and we should not be doing that? And yet you're wondering about the environments where these rinds were and whether or not that's potentially an issue.
Great. So I think it's a really interesting question. Cheese and other fermented foods are an incredibly rich source of living microbes and all of the metabolites they produce. So there's definitely a lot of research in terms of yogurt, which are the lactic acid bacteria, which you find inside the cheese. There has been some research, sort of early stages, showing that microbes on the rind, can survive the digestive tract. And we just had a collaboration where we worked with the Human Gut Microbiome Lab to look at the passage of some of these rind organisms through the digestive tract. And they survive. If they're doing anything, if they have any impact, we don't know. I think it is a really interesting area of future work.
I would just add that that's where all the work is. So I always taste the rind because it is a major contributor of character to the cheese. And I don't always eat the rind, but I always do taste it because there's a lot of work there. Yeah, and a lot of the flavor,
as I talked about, all these microbes are growing on the surface and producing all these interesting flavors. So often if you eat a cheese from the center towards the rind, you'll have this sort of gradient of flavor. So it's often most intense right under the rind, which sometimes is good, sometimes is a little too much, but it's sort of a nice experiment to do when you eat a cheese.
The question So a very interesting talk by both of you, and I guess my question is to both of you. It seems like we have a very chaotic system, and yet you're able to produce the same cheese more or less every time. So it's like there is an attractor towards a certain funneling of the process. And from the model you're suggesting, it seems that there are several key factors that contribute to that. So if you can elaborate a little bit about that.
Yeah, I guess I can speak from the perspective of a microbiologist, which is that during the cheese aging process, there are conditions that microbes really care about that are being fairly tightly controlled. So pH, salt, moisture, and temperature. Those are all things that are important for determining what types of microbes grow, and finally your community of microbes. So we've been doing a lot of research looking at diversity across cheeses from around the world, and it does seem like these conditions that the cheesemakers are setting up are really a big driving force of what type of community you end up with.
Selective pressure. It's all about selective pressure. But the other side to that is there are lots of microbes in cheese that aren't added to the cheese.
Right.
You know, they're microbes that even in pasteurized cheeses that are made with pasteurized milk, there's all kinds of Halomonas is a dominant species in Bloomy rinded cheeses, and we don't add any Halomonas into the pasteurized cheese that you're going to eat out here. It's a cheese called Moses Sleeper, yet it's the dominant species on the rind, and it's probably coming from salt, right? So there's all these other vectors in the environment and elsewhere that are really contributing in a way that, as a cheesemaker, you may have some control over, but
how it works, yeah, we're still starting to understand it, and I think it's even more remarkable that you have these reproducible communities forming, given that the potential for chaos in the system. So I think that's part of what makes it such a fascinating ecosystem to study. What is it that gives us these reproducible communities? Is it just the environment? Is it something about the interactions between species? Those are the sorts of questions that we're looking at in our lab.
Hi, I have a question from online, and it's from Dr. Leonardo Giordano from Colombia, South America. And he is asking, is there any part during the cheese making process where the microbes can turn toxic?
So there are pathogens that are associated with cheese, although pretty infrequently. So things like Listeria and E. coli, Staphylococcus aureus, can potentially contaminate cheese. So that is an issue of food safety.
And in that vein, I think Staph and Staph aureus specifically is probably the microbe that's should be of most concern to cheesemakers out there. Right now, regulators are really fixated on Listeria, but in the cheesemaking process itself, Staph aureus probably poses the greatest risk.
In terms of pathogenesis, or in terms of also what it does to the cheese flavor and such?
The Staph aureus itself is not toxic, but if it is able to grow to certain populations, it produces an enterotoxin, and that can happen even pre-pasteurization, right? So you can have this growth in your milk, pasteurize it. There won't be any more Staph aureus there, but the toxin itself can survive the pasteurization process. So it's likely in Europe it's the pathogen of most concern for cheesemakers, and should be here as well.
Hi, I have a question. Considering the diversity of cheeses that seems to be so easy to make, how come so many different, not so easy. But possible.
Yes.
So many different cheeses are possible from the same limited number of ingredients. I'm wondering why all over the world you get cheeses that are so regionally specific. This one region only produces this type of cheese when perhaps they could produce a lot of different types.
That's a really good question, and it's a question that I wish I could get Dr. Paul Kinstead, who's sitting back here and has done a huge amount of work and research on this subject, to come and answer. Is that all right? Maybe
sure.
not.
Paul, if you want to come up.
You want to talk about that? Because there are major cultural precursors, and history is just full of these technological innovations that have happened that have led these like big leaps in cheese-making technology, and they tend to be regional, but there are also climate-related issues, and the microbiology of different places. You look at like Cork and Irish cheeses, and the Bloomy rinded cheeses of Normandy, which were developed because they're close to a major marketplace, Paris. And then you've got mountain cheeses that are large format, where you had to basically make these giant cheeses and keep them up in the mountains all year and then carry them down the mountain. So I think that to a large extent, that regionalization was really a strategy for making a living as a cheesemaker, first and foremost. But there are these technological leaps, and I think the Celts are my personal like cheese heroes, because what the Celts did for cheese technology really like set the world that we live in on the path that it's on, I would say. Yeah?
I just want to comment a little bit on the Staphylococcus aureus. In historical times, 30s, 40s, staphylococcal poisoning was known as cheese poisoning. Staphylococcus aureus is a heliotrope, so the environment of cheese with the salt and everything, it will favor the growth of Staphylococcus aureus if the temperature is too high. Same thing happened in fermented sausages in the early 70s and late 60s. But what I wanted to ask was about raw milk cheeses. People advocate raw milk cheeses are so much, flavorful and diverse and blah, blah, blah. So microbiologically, do we, what are the unusual bugs that may pop up in a raw milk cheese? Are some of these non-culturable but viable organisms? And how about some of the gas chromatography and liquid chromatography of these raw milk cheeses that might distinguish them from the ordinary cheeses that we use with multiple cultures?
Yeah, so big question. So what are the species that we might see in raw milk cheeses that we don't see in pasteurized milk cheeses? So there's definitely a greater diversity of species in a raw milk cheese. A lot of that diversity is actually in the lactic acid bacteria themselves. So the naturally occurring lactic acid bacteria that are coming in from the milk. But there's a plethora of other species there that you might not see in other cheeses. And those lactic acid bacteria can have a huge impact on the flavor of the cheese, especially not just in the acidification process, but during the aging of the cheese, they can grow inside the curds and create a lot of flavors. So that can be a contributor to the final sort of characteristics.
There are these consortiums of microbes that are associated with raw milk, and they're being reproduced in labs to be reintroduced to pasteurized milk in order to try and recreate, kind of the soul, right? To give these cheeses soul. But Brevibacterium, Microbacterium, cariniforms, in combination with different yeasts, seem to create, you look at the pathways and the behavior of these microbes individually, and they don't seem to contribute that much, but you put them together and suddenly you get an explosion of flavor. And that's really the complexity here, is figuring out how these communities work and what the pathways for flavor production are. And it's a lot it's a lot easier to happen upon that naturally through like the production. You're making raw milk cheese, we produce a lot of raw milk cheese. We have very intense milk quality and screening program in order to prevent cheese sickness. Is that what you were calling it? That's a big problem, and I'm glad we're past that in this day and age, hopefully.
So we had a conversation starting on chaos in the bacterial or fungal side of the cheese, but you also have the whole chaos of the milk side. How do you as a commercial producer control that side of the chaos? Because you can think of the variability over the course of the season, depending if you get from farm A or farm B and how it's going to affect it over time. How do you?
With trepidation, a good amount of trepidation. But there are some fundamental things that you can do. And earlier I said that the farming practices inform the microbial community, right? So we feed our cows dry hay ration, right? We don't feed any fermented feed on our farm. That changes the microbiology in our barn radically. We don't have, we've never found listeria in our milk because it's not something that's growing in the feed bunk, right? We bed our cows a certain way. Our milking protocols are very specific to create the microbial community that we're looking for. In Comté cheese, there's no sanitizers allowed, okay, on the farm, in the cheese plant, or in the caves. Seasonally, depending on the temperature outside, they only wash their milking equipment once a day, and they just flush it, in the morning, or rather in the evening, with scorching hot water. They're selecting for thermophilic bacteria, right? And they're using their pipelines and their milking equipment to, basically grow, as a vector for specific types, communities, right? So depending on the type of cheese you're producing, you need to adapt your practices. If you're just going to go out into the commercial market and take a load of, off a tanker, that's insane. Would never do that, right? It's really about managing practices at the farm level, managing animal health, managing milking protocols, managing sanitation of your equipment in order to end up with the kind of community that you want in the finished cheese. And looking at that community, right? Make sure that the practices are actually producing the community that you're after.
Well, I was thinking more of just the protein content and all the different flavors that you get in the different feed products.
Yes, that's so. Feed is a major component. Feed is going to be a major contributor to flavor production, but the microbes, I would say, are more important even than the feed.
You have to say that in a room full of microbiologists.
Yes, main ingredient.
I have another question from online, and this one is, since the microbial communities of cheese are derived from outside sources, could we use cheese to grow unculturable gut bacteria?
That's an interesting question. I've never thought of that before. The microbes that grow on cheese are very specific, and it's because those are things that can survive the specific enrichment type conditions that you're creating on the surface of the cheese. So things that can tolerate the low pH and the high salt and the low temperatures and the low moisture. So I guess if there were a gut microbe adapted to those conditions, then it might grow in cheese, but otherwise probably not.
And probably not on the rind.
Probably not on the rind, because rind is an aerobic environment. Maybe inside the cheese we can make little guts.
Why are you looking at Why are you looking at me?
All right, right. Well, if there are no more questions, I want to give another round of applause to our
Can I say
yeah,
one
of course.
more thing?
Go for it.
I just can't leave this space without mentioning that the artisan cheese industry is facing a major regulatory threat from the FDA, which wants to ban wooden shelving in the ripening of these traditional cheeses. And I would just encourage you as scientists to ask some hard questions about whether this type of regulation and the regulatory environment that we're living through is going to produce the kind of food that we want to eat in the future. So please maybe just write your congressman and voice your opinion so that we can continue to create products that have existed for millennia and have a place. I believe they have a place in the world that I want to live in. So thank you.
Thanks. So before you guys dash out, I want to let you know we have a special treat this week. If you're interested in learning even more about the microbiology of cheese, our own ASM Press department has just released a new textbook called Cheese and Microbes, and you can learn even more about the history and microbiology of cheese making, quality and safety issues, and the identities of these microbial communities. These textbooks are on sale tonight just outside these doors. They're at a discounted price of $99. And that's not just for those of you who are here in this room tonight. For anyone who's watching online, you can also order one of these books at this special price. If you send an email to books@asmusa.org and you reference this event, you'll be able to order a copy at this discounted price through June 17th. So be on the lookout for that. Also be on the lookout for an announcement about our next Microbes After Hours, which is going to happen probably in either late August or early September. We're always interested in hearing your thoughts, both here and in our online audiences, about ideas for new subjects at other Microbes After Hours. So feel free to email us or tweet those to @microbeworld. And again, I want to thank our fabulous speakers who have just run out of the room because we have another treat for you guys. Matteo has prepared tasting platters of 3 of his cheeses from the cellars at Jasper Hill. So when you guys go out of this room, you'll be able to pick up those and actually get to taste some of the blue cheese, the Bloomy rind cheeses, as well as a washed rind cheese directly from his farm. So thank you to everyone who is able to make it tonight, and for our online audience for watching. Thank you from the American Society for Microbiology. Have a great night.
Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shower.
It's an unusual room, never seen anything like this. Anybody who has access to this facility first has to go through an R scan. So the HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right? Remember, this building is basically a second building inside the main building.